Hello friends, this is Dr. Anjani, Assistant Professor in Biotechnology. Welcome to e-learning platform. Today we learn about structure of chromosome. First we know about the morphology of chromosome. The size of the chromosome is normally measured at mitotic metaphase. These chromosomes as short as 0.25 micrometer in fungi and birds, as long as 30 micrometer in some plants such as trillium. In trillium, chromosomes are very long, about 30 micrometer. But in birds and fungi, it is very short, 0.25 micrometer. However, most mitotic chromosome falls in the range of 3 micrometer in Drosophila to 5 micrometer in man and 8 to 12 micrometer in maize. Like this, the length or the size of the chromosome vary from one organism to another organism. The monocots contain large sized chromosomes as compared to dicotyledonous plant. The organism with less number of chromosomes contain comparatively large sized chromosomes. The chromosome set, the number of chromosomes vary in size. The shape of the chromosome changes from phase to phase in the continuous process of cell growth and cell division. The shape of the chromosome will be changing from phase to phase. So, as the cell growth changes, the shape of the chromosome also changes, and also during cell division. During the resting phase or during the interphase stage, the chromosome occur in the form of thin, coiled, elasticate, thread-like, stainable structure. They are called chromatin threads or chromatin reticulum. But in the metaphase and in anaphase, the chromosome becomes thick, ribbon-like and filamentous. And each chromosome contains a clear zone known as centromere or primary constriction or kinetochore along their length. So, each chromosome, they contain the primary constricted region that is called centromere or the kinetochore. The centromere divides the chromosome into two parts, and each part is called chromosome arm. So, on either side of the centromere, the portion of the chromosome is termed as chromosome arm. The position of the centromere varies from chromosome to chromosome, providing it a different shape. As the position of the centromere changes, the shape of the chromosome also changes. So, the chromosome could be telocentric if they have the centromere at the tip of the chromosome, or acrocentric towards the terminal end, and they have the extension of the chromatin or the chromosome arm. Very short arm will be observed, that is acrocentric, or they may be submetacentric. Approximately around 3/4 length of the chromosome will find the centromere, that is called submetacentric. In some chromosome, the centromere may be located exactly at the center, apparently exactly at the center. Such type of chromosomes are called metacentric. So, the position of the chromosome decides the shape of the chromosome at the metaphase. We will take up one by one. Telocentric, centromere on the proximal end of the chromosome. Centromere at the proximal, at the tip of the chromosome. Next is acrocentric, centromere at the one end, giving it a very short and another long arm. Next is submetacentric. Submetacentric, as I told earlier, will find the centromere at the 3/4 length of the chromosome, and they assume J or L shaped. Next is metacentric. The centromere is located exactly at the center, and the chromosome will be appearing as V shaped at the metaphase. This is the structure of the chromosome. As we know, these chromosomes, when they are stained, depending on the density or the condensation of the DNA, will find the different pattern or intensity of the color. So this is unduplicated chromosome having the two arms. So this is the primary constriction region or the centromere or the kinetochore. On either side you have the arms or the length of the chromosome. These parts are called as chromosome arms. This is unduplicated chromosome. In duplicated chromosome, these are called sister chromatids, and the centromere is the region where the spindle fibers attach to it and they pull the chromosomes towards their opposite pole, and this centromere play an important role in separation of the chromosomes or distribution of the DNA to the daughter cells. This is the structure of the chromosome. It is having a thin membrane called the pellicle, and it encloses a thread-like reticular structure inside that. And the chromosome is not continuous, and here and there you have the constricted region that is called the primary constriction that divides the chromosome into arms. And in some chromosome we will find the another constricted region that is called the secondary chromosome, and this secondary constriction will be having the short extension of the arms or after the constriction, and such type of chromosomes are called as SAT chromosome. They are called as SAT chromosomes. They will be having the secondary constriction. And when you see the closely observe the DNA content of the chromosome, it is not linear. Instead, on its surface it has certain the granular structure. These are called as chromomeres. These are called as chromomeres, and these chromosomes having the tip region that is called the telomere. That is called telomere. So this telomere is very important in maintaining the stability of the chromosome, that prevents the adhesion of the other chromosome, so that it will maintain the stability of the chromosome. This is regarding the structure of the chromosome. And if you observe this the granular region or the chromomere region further, we will find the important components, like it is having the DNA, it is having the protein. The proteins are of two types. One is histone protein and another one is non-histone proteins. So around the histone protein, DNA is wound or it is surrounding, and it will give a the granular nature that is called as the chromomere. If you take up the histone proteins, histones are basic proteins as they are enriched with the basic amino acids like arginine and the lysine. At physiological pH, they are cationic and can interact with anionic nucleic acids. They form a highly condensed structure. The histones are of 5 types called H1, H2A, H2B, H3 and H4. which are very similar among different species of eukaryotes. That means they are highly conserved and have been highly conserved during the evolution. So these histone proteins are cationic and they can interact with anionic nucleic acids. That's why we will find the proteins, the histone proteins like H2A, H2B, H3 and H4, around which we will find the thread-like DNA, which is negatively charged, will wound and it will compactly arrange inside the chromosome. And these sequences are highly conserved during the evolution. Apart from H2A, H2B, H3 and H4, H1 is least conserved, which is its sequence is least conserved among all. Is also loosely bound with the DNA, and this H1 is very important in joining the adjacent nucleosome or the adjacent chromomeres, and this H1 will be having the linker DNA through which the linker DNA passes, and this H1 histone is absent in Saccharomyces cerevisiae. So this is regarding histone protein component of the chromosome. It is having the basic amino acid rich proteins like arginine and lysine. They are positively charged and they will bind to the negatively charged nucleic acid. That's why the DNA is tightly located inside the chromosome, and it is made up of two molecules of H2A, H2B, H3 and H4 molecules. In addition, it also has H1 molecule that helps in linking the adjacent chromomere, and this H1, H2A, these histone protein sequences are highly conserved, whereas the H1 is least conserved among the organism, and H1 histone is absent in Saccharomyces cerevisiae. So after observing the chromomere structure in 1974, the scientist Roger Korenberg described the structural unit of the chromatin, which is called the nucleosome. So these chromomeres structure was studied by Korenberg, and he said that these chromomeres are having the basic structural unit, and these basic structural units were termed as nucleosome. In 1974, he described the basic structural unit of chromatin, which is called the nucleosome. This nucleosome has the 146 base pair of DNA in each nucleosome, and each nucleosome has the diameter of, or it is located within the 11 nanometer distance. So each the nucleosome will be occupied within this size 11 nanometer, and this is having two molecules of H2A, H2B, H3 and H4, which are rich in arginine and lysine, and linker DNA will pass through the H1 that is not highly conserved and may be absent even in some organism. Apart from the histone protein in the chromosome, we also find non-histones, and non-histone proteins are involved in the range of activities including DNA replication and gene expression. So non-histone protein play an important role in the gene expression and DNA replication. They display more diversity, are not conserved like our histone molecules. Non-histones are not conserved. They may also differ between different tissues of the same organism. So non-histone proteins are less conserved and they may differ in different tissues of the same organism, but they play an important role in DNA replication and gene expression. Next is, when we stain the chromosome, they show different intensity of the color, as we saw in earlier slides, and these are termed as Euchromatin and Heterochromatin region. lightly stained regions in the chromosome, when stained with the basic dyes, are called euchromatin. The lightly stained region of the chromosomes are called euchromatin, contain single copy of genetically active DNA. Single copy of genetically active DNA. The extent of the chromatin condensation varies during the life cycle of a cell and plays an important role in regulating gene expression. The extent of chromatin condensation varies with the stage of the life cycle and play an important role in the gene expression and regulation of the gene expression. In the interphase of the cell cycle, the chromatin are decondensed. Loosely arranged and known as euchromatin, leading to gene transcription and DNA replication. For DNA to replicate or duplicate, or in the transcription, we need euchromatin state of the chromosome. So the copying of the sequence will be very easy. Next is heterochromatin. The word heterochromatin was coined by Emil Hertz based on the cytological observations. They are highly conserved and ordered areas in nucleosomal arrays. About 10% of the interphase chromatin is called heterochromatin. 10% of the interphase chromatin is called heterochromatin and is in very highly condensed state that resembles the chromatin of the cells undergoing mitosis. Only 10% of the interphase chromatin is heterochromatin, which will take up more stain. Chromosome contain a high density of repetitive DNA found at the centromeres and telomeres form heterochromatin. So where this heterochromatin region is located in the chromosome? At the centromere and at the telomeres. And in this region we will find repetitive DNA. The same sequences will be repeated many a time. So such type of length of the DNA, they are called as repetitive DNA. This heterochromatin are of two types. One is constitutive and another one is facultative. One is constitutive and another one is facultative. So as the name suggests, if you take up the constitutive heterochromatin, these are the regions that remain condensed throughout the cell cycle are called constitutive heterochromatin. So all over the length of the cell cycle, this region will be highly condensed, remain condensed throughout the cell cycle. Such type of heterochromatin is termed as constitutive heterochromatin. Whereas the regions where heterochromatin condensation state can be changed, so it will not permanent, it will be varying depending on the stages of the cell cycle. Such type of the heterochromatin are known as facultative heterochromatin. So heterochromatin all over the length, the heterochromatin region irrespective of the cell stage and the cell cycle, it is remain condensed. Such type of heterochromatin is called constitutive heterochromatin. If the heterochromatin region relaxes and condenses depending on the cell stage and the cell cycle, they are called as facultative heterochromatin region. Next is telomeres. Telomeres are the region of the DNA at the end of the linear eukaryotic chromosome. So the tip region of the chromosomes are called telomeres, are required for the replication and stability of the chromosome. These telomeres are very important to maintain the stability of the chromosome or integrity of the chromosome. McClintock noticed that if two chromosomes were broken in a cell, the ends were sticky and end of one could attach to the other and vice versa. He took a cell where she never observed the attachment of the broken end to end of an unbroken chromosome. That means unbroken chromosome having intact telomere did not show the attachment of the chromosome at their end, suggesting that the end of the chromosomes have unique features. So the telomere of the chromosome, if it is intact, will maintain the stability and it will give the integrity to the chromosome. The telomere sequences remain conserved throughout the vertebrates and they form caps that protect the chromosomes from nucleases, that protect the chromosomes from the digestion from the nucleases enzyme and other destabilizing influences. How they maintain the stability of the chromosome? They prevent the digestion of the chromosome at the tip and also the other destabilizing influences, and they prevent the ends of the chromosome from fusing with another one. So they prevent the fusion of the adjacent chromosome, and these telomeres play an important role in end replication problem. So this will be avoided by telomerase enzyme, which was identified in 1984 by Elizabeth Blackburn and Carol Grieder of the California University. So these two scientists, they identified the telomerase enzyme, and this play an important role in avoiding end replication problem. So during copying of the DNA sequences, the part of the chromosome or the DNA sequences will be lost. So that is called as end replication problem. That will be prevented by the activity of this telomerase enzyme. This is regarding the structure of the chromosome. One more thing is this primary constriction or the centromere region. The number of centromere will be varying from one organism to another, or else the number of chromosome, the centromere may be one in the one chromosome or maybe two, like that. So that means the centromere number will be varying, and this centromere is having the protein that is kinetochore. It helps in adhesion or attachment of the spindle fiber during the cell division and pulling the DNA content equally to the opposite poles. So that takes place due to the presence of the protein, and they help in distributing the genetic material equally to the daughter cells. I hope you understood the structure of the chromosome. Thank you.